All right, here is our final video lecture in the Unit 6 of our nice happy AP biology course. And in case you're watching and looking at that cartoon and thinking, wait, is that really the sixth step in a procedure? If you have, if you get lab experience and you are actually trying to work these things in the lab, and then you are um, doing this over and over, and a lot of times for every like good result you have, you might have like a good 10 results where you don't see anything, or it seems like the PCR reaction or whatever doesn't work. You do kind of want to go cry under a table sometimes. So I thought this was a nice, accurate view of how science can be sometimes, and a little bit of humor. So let's go now into complicated, slightly scary diagrams. A lot of what we're going to look at is going to be DNA technology, ways to manipulate genes, ways to manipulate what organisms are expressing specific genes. And so you're going to see a lot of references to DNA and things done with DNA. But at the heart of a lot of these technologies is something called recombinant DNA. Recombinant DNA is some sort of DNA molecule, usually made in a laboratory, that is going to be a combination of genetic material from two or more organisms, often two or more species as well. And so the you generally have a couple of different types of DNA. You'll have your donor DNA, which is where you're getting your target gene from. And then you'll have some sort of vector. And a vector is going to be whatever is going to deliver that donor DNA into your host cell. There are several requirements for being a vector. It has to be something that can replicate on its own. A lot of times we're going to have something called a genetic marker in there to make sure that we know and are able to identify when a cell has actually had this DNA introduced into it. And so we've got a couple of things we can use for that. There are some artificial chromosomes, but the most common ways to introduce DNA into a cell and try and get it to express another gene is to use either a DNA plasmid or a virus. So what you see on this diagram is a DNA plasmid. It's a ring of DNA. It's very small, much smaller than a normal bacterial chromosome, and therefore much, much smaller than a eukaryotic chromosome. So you basically digest the DNA with something called a restriction enzyme that will break the DNA at a specific sequence. Here we'll see that for this example, where it sees AATT, it will break that sequence there. Notice now you have these what are called overhangs that are complementary to each other. And these overhangs can then allow what's called annealing, or you can say ligating, will allow these fragments of DNA to combine with each other. And so then, once they're base paired with each other, you get some ligase to come in, seal up any breaks in the sugar phosphate backbone, and now you've got this plasmid, which has your target gene introduced into it. And then if you can get that plasmid into a bacterial cell, now you've got your target gene introduced into a bacterial cell, and then that plasmid replicates on its own because plasmids replicate independently of the main bacterial chromosome. So you can have multiple copies inside of a single bacterial cell, and then each time that bacterial cell divides, it will have at least one copy of that plasmid in it. And so soon, depending on how long it takes that bacterium to multiply, within a few days, you can have thousands, hundreds of thousands of bacterial cells, each with that target gene in it. And then what you do from there kind of varies. But I talked about digesting or breaking down a section of DNA, and that's done with these special class of enzymes called restriction enzymes. So these were originally discovered in DNA, in bacteria, and that's where we find most of them. And this is a kind of system or set of enzymes that bacteria use to try and defend themselves against viruses. So basically, when a virus is infecting a bacterium, it's going to insert DNA into the bacterium. Well, restriction enzymes are there to protect the bacterium from that viral DNA. So what it will do is it will chop up that viral DNA into little bits so that it's no longer functional. Well, we've used those, purified those, and are able to use them to digest DNA of any species because, DNA is DNA. So an enzyme, a restriction enzyme, as long as you have a specific DNA sequence which differs between different restriction enzymes, as long as you have that specific DNA sequence somewhere in your piece of DNA, the restriction enzyme will cut at that specific location. So we've got a number of different restriction enzymes that we can use in the lab. They tend to be named for a couple of things. The big one is the name of the bacterial species that they come from. So EcoR1 is a very common one. It comes from E. coli. ND3 is named for the bacterial species it comes from. I don't remember the name of it off the top of my head, so we'll move on to look at more stuff at restriction enzymes. So a restriction enzyme has two active sites that are basically going to cut both strands of DNA simultaneously. So this restriction enzyme here is cutting here and cutting here. And once it's cut, it basically leaves it can leave these open ends depending on the specific restriction enzyme. There are some other restriction enzymes that will just cut at the same place. So another restriction enzyme might cut here, and it'll make two basically blunt-ended fragments of DNA. But what you ideally want to use if you're doing this in a lab is you want some sort of restriction enzyme that will leave what we call sticky ends. These sticky ends have some exposed base pairs, and these exposed base pairs, or these exposed nitrogen bases, can then base pair with the complementary sequence so you can attach in and stick in other segments of DNA. It's like you take a piece of paper, cut it in the middle, tape more paper inside so you can make a giant piece of paper. That would be kind of like what we're doing when we're using restriction enzymes to make recombinant DNA. So once you have digested your DNA through restriction enzymes, you can do a couple of things. One of the more common techniques that you'll use in a lab is gel electrophoresis. There are multiple types, but we're going to focus on the type that is used to separate out fragments of DNA. And so once you've cut a sample of DNA with restriction enzymes, you can use gel electrophoresis, which works because of a couple of things. One, DNA is negatively charged. So since it's negatively charged, you can run a current through, a chunk of DNA, and DNA will move towards whatever the positive pole is. And so a gel will always have a small electric current running through it, and I'll have a positive end and a negative end, and you have to make sure that you're keeping track of where you have your DNA because it's going to be pulled towards the positive end. If we had had a chance to do these in gels in person, I would really be able to demonstrate this, but we're just trying to get some of the theory down. And so another thing that is a factor is if you look at this diagram, you see what's called a porous matrix. If you look at the actual 3D structure of a gel, what you'll see is that it's got a bunch of fibers and stuff in it that form this kind of matrix that has some holes that will allow DNA to pass through. But it's just kind of like if you're, like, let's say, trying to pass in a really crowded hallway, you're trying to get past people. It's going to be, if you're a really large person, like you're 6 feet tall, giant shoulders, it's going to be a little harder for you to get through. So like I went to Skyline for high school, and Skyline has a lot of kids there. When it was a passing period, it would be very difficult to get through, except I had a bit of an advantage. I'm short, so I would basically kind of duck down and I would move kind of in between people. And since I was shorter, a little smaller, I was able to kind of weave my way through. So I was actually able to move through the hallways at Skyline much faster than some of my taller, broader friends. DNA kind of has that same pattern of movement in a gel. So if you have a small segment of DNA, it's more likely that it can fit through those spaces in the matrix and be able to move faster and move farther in a gel. On the other hand, a very large piece of DNA is going to be kind of stuck, probably closer to its original location. So what you see is this is kind of the sample that you have put into a gel. You run the current So that way there's a positive charge down here. DNA is attracted to that positive charge, but the things that can move the farthest and move the fastest are the smallest segments of DNA. So that's why you see the smallest segments of DNA are found here. Whereas the largest segments of DNA are still pretty close to the top. So you can separate out DNA by its size, and it's a fairly accurate way to do this. So here's just a couple of images showing you the setup. So here is on the left, what you have is you have an agarose gel. You have these little wells. And what you do is you will take your sample and you'll take a pipetter, which if we see an image of it somewhere here, I'll show it to you or point it out. And you basically inject a tiny amount of the sample into whichever well you want to look at. Usually you're also going to include something called the DNA ladder, which has known weights of DNA. So that way you can look at, like, let's say you know that this is, right here is 100 kilobytes. And you know, this here is like 10 kilobases. Sorry, I don't know why I said bytes. You can kind of see if you've got a DNA band that lines up very similar to that, you know its approximate size. And so what happens is you run a charge, DNA gets drawn down this gel. The smallest pieces are going to move fastest and they're going to move furthest. And as this gel runs longer, you get more separation between the different bands. Here, what you're seeing is another diagram showing you very similar thing. You know, you've got a charge running through, there's a positive charge. You've got your samples basically being inserted into these wells. Everything's sitting in what's called a buffer solution that just lets the charge pass. And then what you can do is you could take the same DNA sample and you could add 3 different restriction enzymes, or 2 different restriction enzymes and then a combo. And these restriction enzymes, if they cut at different sites, will make fragments of different lengths. So what you see here is if you use enzyme 1, you're going to get fragments A and B. If you use enzyme 2, you're going to get fragments C and D. Notice they're all different sizes, so they should separate out differently. And if you use both of those fragments, you'll get fragment A. I mean, both of those enzymes, you'll get fragment A. But then that part that was B ends up getting cut in half to make fragments E and D and E. And if you'll look, the shorter fragments, because D is pretty small, are going to go the furthest. They're going to travel the farthest. The next shortest fragments, which are A, will be next. Then you're going to have the next shortest fragment, which is E. The next shortest fragment, which is B, and then finally the longest fragment is furthest back, which is C. So you see these kind of characteristic patterns of bands. And then a great thing is this diagram also has an actual picture of a gel. Usually you stain it, so that way you can visualize all of the bands of DNA. You have some sort of pigment molecule that will attach to DNA, and then you'll be able to see this pattern. So you get 3 different types of information from a gel when you run it on DNA. You'll get information about how many fragments you get. So depending on what restriction enzyme you use and how many times the site that restriction enzyme recognizes occurs in your segment of DNA, you'll get, 2 fragments, 3 fragments, 5 fragments. You'll get the sizes. So you'll have what's called a ladder. So this is probably a ladder, and this is probably a ladder. You tend to put ladders along the edges, so that way you can tell, like, oh, okay, I know that's my ladder. And though you'll have, depending on where you get your ladder, you'll usually get a set of instructions with it that will tell you the sizes of the different bands. And so then you can use that to estimate, okay, if I know what size this band is, then I should be able to figure out the size of this band because it's around the same place. And then you'll get an idea of relative abundance. So for example, here you see this very light band. There's not very much of that DNA fragment. However, here you see a very thick band. That means you've got a lot of fragments of that size. So those are the pieces of information you can get. So just showing you again, what you see is that the bigger the DNA, the less distance it migrates. Here is, for example, a ladder, like I called it, kind of because it looks like it has steps like a ladder. And then, so for example, if you have one marker here at 1,000 base pairs, any place where you see a marker at the same location or same distance from what is the well, is going to also be 1,000 base pairs. Just like anything that is over here is also going to be 200 base pairs. So you have a band at 200 base pairs. There will be some bands that may not line up exactly. Like this one right here is somewhere between 400 base pairs and 500 base pairs, but you know it's closer to 500 base pairs because it's closer to that line. Once you've separated out your fragments of DNA using the gel, you can actually cut out chunks of this agarose and then you can melt it and then you can purify out that DNA. And then if you want to take that DNA and then, attach it to a plasmid, or you can amplify it using PCR, which we'll talk about in a moment. Now, gel electrophoresis has multiple applications, one of which is in forensics. It's usually not quite this simple anymore. There's some more complicated analysis that goes on, but this is kind of the most simplistic way you could use a gel to analyze DNA to try and see, okay, who is responsible for this crime or match up victims, etc. So what you see here is you see a sample from a crime scene, and then you see samples from 3 different suspects. Now, these have all been digested with the same restriction enzyme. If you see the same pattern of fragments, then you'll know that, hey, these must have come from the same source. So if you have that sample of DNA, let's say it's from some blood splatter, that was found at the crime scene and it matches, once it's been digested, the band pattern on your gel matches that of one of the suspects, you've got something tying them to the scene, because the likelihood of any of the other suspects having that same kind of a band pattern when digested with the restriction enzyme is very small. Now, it's not 100% foolproof, but, a lot of people have actually been exonerated by DNA evidence. So let's talk about the next type of biotechnology, polymerase chain reaction, or PCR. And it's a really big shame that we were not able to have class this year, because I have PCR machines that we were able to acquire, and it was really cool to be able to use these. So what PCR is used for is to amplify small samples of DNA. So let's say you've got like 20 copies of DNA or 30 copies of a specific region of DNA, but you need a lot more than that, because you're going to try and do some bacterial transformations. You're going to try and insert that gene into a plasmid or a virus. Or another thing is you're trying to figure out, you're trying to like do some gene sequencing. Well, you can do that if you use PCR. PCR is basically a way of making thousands, millions of copies of a targeted sequence of DNA in a fairly rapid amount of time. So, one of the quicker PCR cycles that we have on our machines is, I think it runs about an hour and a half. So after an hour and a half, you'll have millions of copies of a specific DNA fragment. What we would do is we would take our own DNA, so we'd take some cheek cells, get a swab of them, would digest them with certain restriction enzymes, and then run them PCR, and then run them on a gel, and we were actually able to see if we were homozygous or heterozygous for a certain trait. It was really cool. So this is kind of how PCR works. You have a tube, and you're going to put a lot of stuff in it. You've got a mix of primers, so it's going to be basically these small sequences that are going to kind of match up to certain regions of DNA. You'll usually just have one primer per PCR that you're running. You're going to have a specific type of DNA polymerase that is heat resistant, so you can heat it up to, say, 95 degrees Celsius, and it's not going to denature. But your DNA strands will separate. And then you're going to have a bunch of nucleotides in there. So it says DTTP, that's D thymosin triphosphate. That's a DNA cytosine triphosphate, adenine triphosphate, and guanosine triphosphate. So you've got the building blocks for making DNA. What you're going to do is you're going to heat these up so your two DNA strands separate. These primers will attach to whatever sequence they're complementary to, and then this polymerase will come in and it will start building a new DNA using these nucleotides, starting from the primer. Because remember, you need a primer for polymerase to attach, and then it will start building you some new DNA. It's going to do this on both strands, both you could say this is your template strand and this is the complementary strand, and so you're going to end up with, from a sample, one molecule of DNA, now you've got two. This cycle gets repeated like 20 to 40 times. You then heat things up, strands separate, primers attach, the polymerase comes in, builds new DNA molecules, then again you heat it up, strands separate, primers attach, new DNA molecules are built with the polymerase, and you do this 20 to 40 times, depending on the sequence that you're trying to get replicated, and this makes a lot of DNA. So what do we use this for? Well, it is a very specific process. You pick a primer that is going to match whatever sequence of the gene you're looking for. So I've actually done this in a lab. I did this with fruit flies, and we sequenced the entire fruit fly genome. So I was able to look for my target gene because we were trying to knock out a gene. And I was able to look, like 50 bases upstream of it. Choose a segment, use it as a primer, send off to a company. They would make the primer, so make those segments of DNA, ship it to us, and then we were able to use it in PCR, and we're able to amplify these segments of DNA, which could then be used to kind of take the gene, make more copies of it, take a mutated version of the gene, make more copies of it, and then be able to insert that gene into other fruit flies. You can also use this to make lots of copies of genes, of segments of DNA, which include a gene that you can then digest and use as recombinant DNA to, insert into a vector. You can actually use a process with PCR with something called reverse transcriptase. And if you look, the ACE is an ending that should let you know it's an enzyme. And I'll talk about that on, I think it's the next slide or so. You can actually take all the mRNAs that are being expressed in a cell. You can use reverse transcriptase to make DNA copies, and then you can use PCR to make a bunch of copies of those genes, and you can actually figure out what mRNAs are turned on in a specific cell at a certain time. You can also use these to find what are called single nucleotide polymorphisms, or SNPs, or abbreviated SNPs. And these are these useful genetic markers. So a lot of times when we're looking at, you have this version of a breast cancer gene, or you have this version of the hemoglobin gene, we usually are looking at, instead of the exact gene, we're looking at these single nucleotide polymorphisms that are located next to that gene. They're really good genetic markers, and that's kind of where we tag a lot of, gene variants that we see. We're like, okay, it's this gene, it's next to this SNP. Okay, so we're going to look for this SNP in our PCR. So reverse transcriptase is actually an enzyme that we stole from viruses. There's a class of viruses called retroviruses. Retro means backwards, and that's because this process kind of runs backwards from normal. It's called reverse transcription. So you know how transcription, you go from DNA to RNA? That's transcription. Well, the reverse of that, reverse transcription, is when you start with an RNA molecule and you make a DNA copy from it. So what ends up happening is retroviruses like HIV, human immunodeficiency virus, actually have their genetic material as RNA. When it gets put into a cell, they actually dump in their RNA, and they also dump in this reverse transcriptase, which then uses their RNA as a template to make some complementary DNA, which is the stage that you see right here. So here's reverse transcriptase. It's basically taking this RNA and it is building a complementary DNA, which is this top strand. Then what can end up happening is that reverse transcriptase can then run back over it again and replace the RNA with DNA. So then after that, you're going to have double-stranded DNA. So you can use that for RT-PCR, which is reverse transcription, transcriptase PCR. That's actually really important for right now because with the COVID-19 pandemic, you may have heard of PCR tests to test as accurate tests for seeing if somebody is infected with COVID-19. And you may have had a COVID test done where they stick a swab really far up your nose. So far, it feels like they're trying to scrape your brain off. What they'll do is they'll take that swab, they'll, stick it as far up your nose as they can because they're trying to go where the virus will usually gather when it is present in a person. They will then put it in a solution, which, if there are viruses there, cells, whatever else is in there, basically the DNA is going to be extracted from it. Then you use reverse transcriptase to turn the RNA into DNA. You have to do that because things like COVID-19, coronaviruses are RNA viruses. Then you're going to run you're going to run a PCR to amplify the DNA that's made. This is something that you can actually do in an RT-PCR machine now. And then the primers that are on there have fluorescent probes or dyes so that you can visualize them. And then you've got, basically, using this dye, you can detect if that dye is present then in the sample, then you know that, okay, you've got some viral RNA that was in that sample, which means you had copies of that virus running around up your nose. So this is a much more efficient way. These are kind of the really accurate tests for COVID-19. And the fact that we have all of this technology, reverse transcriptase and all of that, really helped us to be able to try and diagnose COVID-19 a little better. So let's talk about another experiment we would have done if we had had class, and that is bacterial transformation. So when you transform bacteria, you are basically introducing other genes into a host bacterial cell. So usually this is going to involve a plasmid. So here you have a plasmid. I think this one is P-Flow. It's got a gene for a fluorescent marker. So any bacterial cell that has that plasmid in it should glow. Usually under like UV light or something like that. But what you do is you basically treat these bacteria in a way that makes them more likely to take up a plasmid. And then you see that plasmid right here. Any bacteria that have that plasmid, usually you'll have some way to weed those out from the bacteria that didn't get transformed. And these bacterial cells that are modified are now transgenic cells. That's what you call organisms that have had, their genes changed. So how do you know if that bacterial cells actually got transformed? Well, what you do is you put some sort of selectable marker in the plasmid. A very common one is antibiotic resistance. So there are genes that bacteria can pass around to each other on plasmids that make them resistant to antibiotics. If you do that, if you say, okay, I'm going to make sure that if you have this plasmid in you, then you've got a gene that makes you resistant to ampicillin, which is an antibiotic. If you grow these bacteria on a plate where the growth medium has ampicillin in it, any bacteria that grow have that resistance gene and therefore took up your plasmid. And you know that because if they didn't take up the plasmid, the ampicillin would kill them. So that lets you tell, okay, these are my transgenic, my transformed bacteria. You can pick those bits of bacterial goop off the plate, and then you can let those grow somewhere. There are also now, technology improves, and instead of just using like antibacterial plates, what you can also use are reporter genes. So the two most common ones, one that's been around for a long time, is beta-galactosidase, which is a LAC-Z gene, and it codes for an enzyme that if you have a white substrate in this certain chemical in your bacterial food, it's going to turn that into something that's bright blue. So any of the blue colonies are going to be the ones that are transformed. Well, somewhere around the time when I graduated from college, scientists discovered green fluorescent protein from jellyfish, and they were able to sequence the gene, and now they use it all over to make glowing things. So, what we would have done if we'd been able to do the experiment is we would have transformed some bacteria with a GFP reporter gene, and then we would have been able to see that we had that gene because we would have seen glowing colonies. They glow when they are exposed to UV light, so then we would have known we had successful transformation. Don't feel too bad. We actually haven't had much luck with this. Okay, so genetic modification and cloning. Gene cloning is you basically take a gene of interest from whatever organism that you get that gene from. Then you can take that gene, stick it into a plasmid, and use it to transform or transfect a host organism. So here we've got, this is a obviously eukaryotic cell of some sort. How do I know that? Well, it's got a nucleus. Here's a prokaryotic cell. I know that from multiple ways. One, it's a bacterium. One, it has no nucleus. And basically, I can take a plasmid, extract my gene of interest, stick it into the plasmid, the way you make recombinant DNA, stick it into the bacterial cell, and now, when the plasmid is replicated, I'm going to have more copies of that gene. And when the bacterium reproduce, I'm going to have a lot of copies of that gene. So there are multiple ways that you can introduce recombinant DNA into cells. So if you're not a bacterium, it's a little more difficult to get DNA into, but there are a few things. One, you can use a specific species of bacterium to insert DNA into plant cells. You can make transgenic animals by taking a very, very fine tip needle and injecting DNA into the nuclei of fertilized egg cells, which when they develop into an embryo, most, if not all of those cells should then have your recombinant DNA. There's actually something called a gene gun, which shoots host cells with tiny bits of DNA. When I first heard that, I was like, wow, we really will use guns and shoot anything. And then you can also use viral vectors, so a virus that you can, take out any of the dangerous genes, put in your desired, gene of interest, and then you can use that to especially introduce this gene into eukaryotic organisms. Which is great because a lot of eukaryotic genes are too big for plasmid. They don't fit. So, one thing that you can do with this is you can take bacteria or yeast cells and you can introduce a gene to them. And that gene, whatever protein it produces, can be reproduced in that DNA, in that bacterium with that plasmid, grow a bunch of these bacteria, have them all make this protein, and then you can make a bunch of copies of a protein from bacteria. This is actually how we got insulin to go from being a very expensive, hard to get drug to being something that's very easily available. Back in the day, you used to have to kill a pig and grind up its pancreas and isolate insulin from it in order to get insulin that people who have type 1 diabetes could inject. Now we actually have two and we'll have two strains of bacteria. One will have one subunit for the insulin polypeptide, another will have the other subunit. We'll stick that into some bacteria. We'll get those bacteria to express that protein, and then we'll basically, you know, use some chemical reactions to cleave those peptides and fold them into their final shape to make a functional insulin molecule. And you can do this on a very large scale because bacteria are pretty cheap, easy, and fast to grow. This is being taken to even further extremes by a process called farming. Stands for like pharmaceutical and farming. Basically, you use farm animals or plants to produce some sort of pharmaceutical drug or something. So what you can do, for example, is stick a transgene, your desired gene of interest, next to the promoter for a protein that's produced in milk. And then if you do that, you stick that into, say, a cow or a goat or a sheep. Then whenever that, you've got to make sure it's a female. Whenever that animal lactates, its milk will produce that protein. So there's actually a set of experiments where people are trying to make a lot of spider silk so they can harvest it. So they actually put a gene for making spider silk into a goat, so that goat produces the proteins that make up spider silk, spider webs, in their milk. It's a little creepy, but it works because it's not like you can go milk spiders. I would not try that. So, there are a number of different chemicals that we can make with farming. So, erythropoietin is it's involved with red blood cells, so you can use it to prevent anemia in patients. I mean, you can read through this, you can pause this video and read through. Human insulin is there. There are all these chemicals, medically useful chemicals that we can produce using farming. So now we're getting into the territory of genetically modified organisms, or GMOs, which you probably hear a lot about. I'm not going to talk about the ethics of them. I'm just talking about what they're being used for. Basically, you take genes for some sort of function, you're going to stick them into crop plants or animals that are raised for meat. So I know I focus on plants here, but you can actually have transgenic animals. There's, I think, a strain of cattle that was produced transgenically, which looks like it hulked out on steroids, but it's designed to overproduce muscle because muscle is generally what we eat as meat. So those cattle produce a lot of muscle per individual animal. So, as far as crops go, you can actually do some genetic modifications to improve the nutritional characteristics of um, a particular plant. So, for example, rice is a grain that's used to feed a large part of the world's population. However, you can actually make it a little more nutritious by adding genes to it, which cause it to produce something called beta carotene, which is a precursor for making vitamin A. If you then grow this strain of rice, which is called golden rice, and feed it to people, you can actually reduce or eliminate vitamin A deficiency in populations that you feed this rice to, because now instead of not getting any beta carotene in their diet, you give them the rice that they were already eating, and now they're going to be able to make their own vitamin A. You can also create crops that are tolerant of different environmental conditions from what they normally are. So, if you take AP Environmental Science at any point, or if you have taken it, you'll learn that we have problems with our cropland becoming saltier and saltier, high salinity. So what some scientists have done is they've taken some salinity resistance genes from mangroves, which grow in like estuaries, which is a mix between salt and fresh water, and they've inserted those genes into rice to create rice that can grow in soil that is much saltier than they normally can. So it doesn't matter that we've, messed up some of our farmland by making it saltier. We can still grow stuff there. And then you can also create plants that are resistant to pesticides. So there's this really big thing um, where a company created what's called Roundup Ready soybeans. These are soybeans that are resistant to a specific pesticide. So, it's a weed killer basically. And you can spray Roundup all on these soybeans. The soybeans will live, any weeds in the area will die. So, you can look at some of these applications of genetically modifying plants. Here you also see a couple of varieties of golden rice. It's called that because of the color. The color is due to beta carotene. You can also make transgenic animals. Transgenic animals can be used as lab models. You can study the function of individual genes. You can also make animal models of human diseases so that you can study those diseases, how they arise, and also how you can treat them. And you can add these genes from organisms from the same species or from a different species. So you can also use this to produce proteins that you want. That's a little simpler with bacteria. You don't got to feed bacteria as much. So you may have seen images of this unfortunate transgenic mouse, which looks like it's growing a human ear on its back. It's not actually. What happens is this was an experiment where they basically made a synthetic cartilage framework for an ear. They implanted that, and then the mouse's cells started growing around that synthetic cartilage framework. Um, and it actually had a purpose. They used this technique that they kind of perfected in the mice, and they were able to use it to treat this one child who was born with no bone or cartilage covering the left side of their chest. So there was nothing protecting their heart. Like if you'd punch them in the chest, they would not survive. But this technique was used to make a synthetic cartilage framework for them to grow some protective covering over their heart, and now it's actually pretty functional. That child has, less fear of getting punched in the chest. You can also make knockout animals where you inactivate a specific gene. When you inactivate that gene, you can then implant that defective gene or missing gene section into an embryo, and you can see how it causes changes in the development of that organism. So there are lots of strains of knockout mice which have had specific genes knocked out or inactivated. And if you see some radical difference in their physical development, you'll know, okay, well, when you don't have that gene, this mouse may have very low muscle mass, or this mouse may um, like tend to become obese very easily. So you can try and figure out what those genes do. A new kind of gene editing that has become really common in the past decade or so is CRISPR, which you may have heard of. It's technically CRISPR-Cas9 gene editing. CRISPR stands for, like, a description of the technique. Cas9 is an enzyme that's used to edit genes. So you're able to very highly you're able to have some highly targeted, highly specific editing of genes. Unfortunately, when you're trying to transform or transfect an animal or a bacterium, it's a bit of a crapshoot if you're trying to get DNA into the host genome, because you can't necessarily control where exactly it gets inserted. However, with the CRISPR, what you can do is you can target the gene that's already in the host genome and you can modify it. So you don't have to worry about, oh, yeah, I added DNA to this mouse and it accidentally knocked out an important gene, and that mouse was not able to live, so I wasn't able to create my mouse model. So what happens is you have a guide RNA molecule that binds to a targeted DNA sequence. You figure out what gene you want it to bind to, you design it with that sequence or a complementary sequence. That then attracts Cas9. That Cas9 is going to attach where that guide RNA is attached, and it's going to cut both strands of DNA. Those cuts in the DNA are going to cause the cell to initiate DNA repair mechanisms, and they are going to start repairing that DNA. In the process, you can introduce a mutation, so you can make that gene ineffective, you can delete that gene, etc., and you can introduce like a functional copy of a gene if you've got somebody who does not have a functional copy of that gene. I believe there's already been one like actual human trial where there was a little girl. This was her like absolute last ditch effort. Her parents gave approval for them to use CRISPR to edit her genes, and I think it was a success, but I'd have to go look it up. So, gene sequencing. This is kind of how modern gene sequencing works. It's a little complicated. If you're interested, we can go into it in more detail, but it basically involves PCR. You're going to amplify whatever segment of DNA. Then you've got these fluorescently labeled nucleotides, a small amount of them mixed in with the rest. And these fluorescently labeled nucleotides are modified so that when they get added in, you can't add anything else to that sequence. Like, for example, this fluorescently labeled guanine came in. Well, it's impossible for the polymerase to add more nucleotides past that guanine just because of the way it is chemically. And so then you're only able to make a very short sequence. Then you can separate out those fragments by a gel electrophoresis, which will then separate them by size. And you can go from the very smallest, which was just this thymine, and you can have different fluorescent markers on the different bases. So thymine can have one, guanine can have another, cytosine can have another, adenine can have another. And then you can read these. And so here they're reading it with a laser beam, and you're able to basically see what, like, okay, the segment that only had one nucleotide, it ended in a T, so it had to have a thymine. The segment that ended in only two nucleotides, it ended in a T. So that means my second base needs to be a T. The segment that only had 3 nucleotides in it, well, it ended in a G. So that means my sequence is TTG. If this is complicated or problematic, let me know. We can look at it. It is not essential that you understand this. You just need to know you get a gene sequence. So, what you're doing is you're figuring out the order of nucleotides or the sequence of nitrogen bases in a DNA molecule. If you do this with like all the chromosomes in an organism's cells, you can actually determine the sequence of the entire genome. And this was a huge project that was done around the time when I was in high school and college. It was called the Human Genome Project. It was a worldwide collaboration. And the first human genome was sequenced in about 2003. Since then, we've had thousands of species genomes sequenced, and there are even some ambitious projects in the works to try and sequence millions of species genomes. So, bacteria, we've got rats, mice. If it's an important species as far as research goes, or like honeybees are on this list because honeybees are very important for agriculture, chickens, dogs, etc., then we've sequenced its DNA. So you can actually look up like a dog genome or a gorilla genome online. Then you can actually look and compare the genomes of different organisms to try and figure out things like evolutionary relationships. You can compare genomes of individuals of the same species. So, for example, you could do this for a group of people, and you could compare the individuals who have a disease, what kind of variants they have in their genome and their genes, versus an organism or a person who does not have a disease. So that is it for biotechnology. Next up is going to be Unit 7, Evolution, and it is my favorite unit, so be prepared for an excited mystery.